Welcome to today's episode. I'm excited to have Nicolas Maignan with us. Nicolas is an industrial engineer and AI expert dedicated to solving healthcare toughest challenges. As Chief Operation Officer at Kantify, he drives innovation in drug research, focusing on cancer and rare diseases, and has been pivotal in developing their drug discovery platform. With over 7 years of experience as a machine learning engineer, Nicolas excels in cutting edge AI, ML ops, and data science, bringing both technical mastery and a visionary approach to his work. Today, we'll dive into his journey, Kantify's groundbreaking innovation, and their work challenging the one drug, one target paradigm. Nicolas, welcome to the show.
Thank you, Thibaut. I'm delighted to have the opportunity to do this podcast with you today. I hope your audience will have a nice time with us and that they'll be interested by this one drug, one target dogma discussion we'll have.
Most people I talked to so far were trained life scientists. You're an engineer by background, and now you work in a life science company, which I think is the best thing to do, to be honest. I applaud you for deciding to apply your skills to this important field. But can you tell us a bit more about your journey, what you studied, and what brought you to what you're doing today? So why, as an engineer, you decided to apply your skills to life science and not something else?
Sure. I realize that I'm a bit of a weird duck in the industry, but I think it fits really well now. So historically, I studied industrial engineering at the French National Polytechnic Institute of Grenoble. So I was specialized in product engineering and what used to be called Industry 4.0, which implied advanced mechanical engineering, design, simulation, programming, obviously, things like 3D printing, prototyping, and management of the life cycle of products. In 2016, I had one of these internships to do. So I applied to a number of companies, including a small startup in Belgium called Kantify. And I met someone called Nicolas Maignan, who has been my mentor ever since. And Nick was super interested in AI already, which for 2016 was really early, if you think about it. It was the early hype of, let's say, AI, mainly in computer vision, that started 2 previous years. And so he had a project with a big retail company. They wanted to build those smart shops. So with the cameras that would detect the products and people grabbing products. He was looking for a young industrial engineer, and a student specifically, to help him set up the grid and write the code to do object detection and the detection of the action of people grabbing things. And that's how I fell into AI. For the first time, really, I learned about computer vision, the classic computer vision, and then AI-based computer vision. I took the very first fast AI course from Jeremy Howard in 2016 when it was coming out. And I quickly realized that AI, machine learning space, had the potential to replace everything I had learned at school regarding industrial engineering. So when I understood that, I made a deep dive. I went into AI fully, and I had the chance of being hired by Kantify at the time, even though I was still a student. So for 2 years, I was part-time engineer, part-time engineering student at school, and I did a lot of consultancy projects. So for the first years of my career, it was mostly consultancy in many different industries. I worked in energy, chemistry, logistics, fintechs, psychology too, and transports. And it was always AI consultancy. So we would come in, the company had a challenge, a business challenge, and we would propose like a machine learning based solution to the challenge. Thanks to that, I had years to learn about all the different types of machine learning, all the different types of data, from image, obviously, to signal, tables, tabular data, as we call it, text, so with NLP. And over time, we felt like we liked a number of these projects, but some of them were lacking, let's say, we didn't have emotional attachment to them. We didn't really care about what we were doing. It was mostly optimizing things, optimizing processes for big industrial companies. It wasn't what we wanted to do. So we put a grid, project selection grid, for the whole team to fill in, to make the decision of which contracts we would take. And one of the criteria was actually ethics. Do we like ethically the project? And when we stored that, slowly over time, all our projects became health projects. We kept working more and more for the health industry. For example, we worked on computer vision for blood pathological object detection, things like that. We were the first ones to be capable of predicting atrial fibrillation several minutes in advance based on onboarded wearable device data. Things like that. And accidentally, someday, we wanted to test some new types of neural networks. We found a very interesting use case in drug discovery. It was like small molecule protein interactions. We took some data to build a model and test these new graph neural networks at the time. And we realized that we had something that worked super well. And that's when we started our drug discovery journey. So this was about 5 years ago now, a bit less than 5 years actually. And since then, we've only committed to that.
If you look at drug R&D, how it's done, it's one of this industry that takes a lot of time, costs a lot, fails a lot. So it's often criticized for being slow, expensive, and with lots of failure, essentially. So what's your take on the process, and why do you think it's broken in so many ways?
I must say that drug discovery, life science, pharma companies, it's such a unique sector to work in. It's different from all the other ones I had the chance to work in, namely in the way it's structured. And I think that's actually the first problem maybe in drug discovery. It's the business logic behind it. So as you said, the drug discovery process is extremely costly. I've read that it's about 2 billion in average investment for a drug that made it to market from start to finish. And the problem is the success rate is extremely low, reported 2 to 4% success rates over the entire industry. Meaning that every investment made has to be measured through time to maximize the chances of success. But sadly, like, it's still an industry that costs a lot, and the return on investment is far from being guaranteed. Sadly, most of the human diseases are rare. There's a couple of indications that are prominent throughout the population, but the 5,000 plus human diseases are mostly rare, meaning that they have very small markets attached to them. This means that there's a very small or low incentive for companies to put 2 billion of investment for a 2% success rate of finding something that can be used in only a couple hundred or thousands of people in the end. So I would say that this is the first flaw of the field, this massive need for investment, low success rate, and sadly meaning that a lot of diseases will never, at least if it stays as it is today, never have an actual drug. That's the first, let's say, business explanation to why our R&D in drug discovery is broken. But the second one, I think, is just biology. Biology is extremely complex. And I believe that our understanding of it today is extremely limited. The state of drug discovery today is more like a reflection of the huge biological uncertainties when it comes to identifying safe and effective drugs. There are many pipelines where the preclinical studies, so before we get into humans, are very complete. Everything has been done. The toxicity of compounds has been studied extensively on different types of animals, the best models to date, and it gets in phase 1 and it fails for toxicity against humans. Still, today, there's a lot of struggle in predicting those adverse effects, which explains a lot of the dropout of drugs during clinical trials. So that's, yeah, I think that the main challenge to drug discovery today, just the sheer complexity of biology.
So you mentioned drug discovery it's an expensive game. There is a very interesting scientific paper that came up with this number, but very recently the same author revised this figure. I think this figure, they computed it like 10 years ago. And they say if you look at the top 16 pharma, so really the largest one, the figure is not 2 billion, it's 6 billion. So it's even worse. Higher. Yeah, it's crazy. But then I think my question is, pharma as an industry is one of the industries with the highest spend in R&D, and this is actually increasing. I was looking at the number from the Pharmaceutical Association yesterday. So it's been increasing a lot in the past 10 years, but we don't really see progress. So when you look at the number of drugs you get per billion of R&D invested, in the 50s, it was around 40, 50 drugs per billion. And now if you look at adjusted by inflation, we are around 0.4 or between 0.4 and 1, depending on the years. So it's really bad. So there is a lot of money, very poor return. And I'd love to get your take on why is it so bad, despite all this investment, we haven't made more progress, or maybe to turn it in a more sarcastic way, no matter how much money we throw at it, we don't really solve the problem.
Yeah, I have a specific take on this. I think that there's a fundamental challenge to drug discovery that is approached the wrong way in the industry. The first thing in drug discovery is not actually drug discovery, it's target discovery. To develop a drug, you need to have a notion, an idea of which biological process you want to modulate in order to resolve a disease condition, a phenotype as we call it. Today, the trend for target identification is that it happens through a lot of omics analysis. So in short, researchers, they observe the state of sick cells in terms of the presence or absence of biological entities like proteins, RNA, so DNA expressions, and things like that. While this technique allows for the identification of biomarkers quite efficiently, biomarkers aren't necessarily good therapeutic targets. Very often, biomarkers are actually like just the last level footprint of the real underlying disruption somewhere in a hidden biological pathway above it. And if you look at a lot of drug discovery pipelines nowadays, they focus on biomarkers. Someone has identified overexpression of a given protein in a cancer and is looking for inhibitors of this protein, thinking that naturally it should counter the phenotype of the cancer. But it's rarely actually working. Usually this is just like, this overexpression is due to several levels of chemical interactions above, some protein disruption, and this is this protein that you should be targeting. So that's one of my explanations to this. A lot of the targets are just the wrong targets in the field. And I have a second thing that I identified as potentially explaining why we have less than expected drugs coming into markets. It's what I would call abusive IP protection. Humanity has identified, let's say, about 80,000 small molecules that we would call the repositionable, a set of all FDA-approved investigational drugs plus naturally occurring small molecules that are all of high biological interest. They've been designed either by humans or by nature to have biological effects. If you look at the patents attached to those thousands of molecules, you'll see that they are widely covered against almost every possible indication, often without sufficient in vitro evidence to back up the claims made on the patents. But this is usually sufficient to block the exploitation by other players. And several times I have encountered partners, for example, patient associations, who have identified that one of these repositionable drugs was super useful in the context of one specific rare disease, but who have been blocked in negotiations by the pharma company owner of the rights of the molecules. And what's crazy is that usually they don't want to spend money just because it's a rare indication, there's no market, but they don't even want to release the rights for exploitation. Abusive IP is, I believe, one of the problems in the industry. If pharmas were slightly more keen to letting others exploit some of their molecules, we'd likely have more drugs to give to people for a number of rare indications. Yeah, that's my second explanation.
Yeah, and obviously this one will take us in an interesting direction. So it's an interesting take because we tend to focus on the scientific problems, on the tech problems, and obviously here it's more legal challenge and a business protection that are, as you said, sometimes maybe abusive. But let's go back to one of the things that we were discussing in our initial call. This maybe outdated perspective that in biology you have one disease, you identify a target. If you can modulate this target, then you will do something to the disease. So this one drug, one target paradigm. And you mentioned that there is this persisting mindset around that, and that's also a little bit problematic because sometimes it works, but most of the time it doesn't. And you might have to look at multiple targets to address one condition. And I think anyone who ever looked at biological pathways, when you see the complexity of biological pathways, that there are many roads that lead you to the same thing. So if you block one road, most probably there is another road to go somewhere. So there are escape mechanisms for disease. Do you want to say a few words about?
Absolutely. Absolutely. So many call it the one drug, one target paradigm. I call it a dogma because it's really almost a belief at this stage. Some people are convinced this is a reality, that drugs are designed to be extremely selective, potent on one target, and that this is what leads to in vivo effects. But there's more and more evidence that it's not the case, and that even making the drugs super potent on a single target and very selective does not correlate with more drugs showing activity in vivo settings. If you look at the pipeline page of most pharmaceutical companies or small biotechs out there, you'll always find the same table. It's a table where the first column is targets. You have a single target name, and then they say we found 10 inhibitors of this target. It's the core of the problem. People are working on single targets. They want to find potent selective modulators of these specific targets, believing that this is modulating just that is going to be sufficient to have an in vivo effect. So while they'll have very successful in vitro assays, functional, then maybe sometimes on cells, as soon as they get in vivo, problems start. Biology is very complex. And as you said, we know that biological systems are networks of interactions. There are many feedback and interaction loops in our protein interaction pathways, meaning that if you block a single thing, there's almost always a redundancy somewhere that will activate and compensate for it. And it's something that is really well observed now in oncology research. For the longest time, there was the race at identifying the best cancer target to be inhibited to create a cancer killer drug. But if you look at the latest trends, the whole oncology field is going towards combination therapy. It's the conclusion is here. If you need combinations for your drugs to work, it's likely because modulating a single target doesn't work. That is, if the existing drugs were actually potent and selective modulators of single targets, but it doesn't even seem to be the case. There was a very interesting study in which a group of researchers took the 300 most common oncology drugs, which had an identified putative target of interest, and then they genetically engineered the corresponding cancer cell line to silence the putative targets. So they removed the supposed targets of these drugs in the cell lines, and they tested the drugs again. Most of the drugs maintained their effect. So they remained cancer killers. Sometimes they've lost a bit of effect. They were slightly less potent, but they still were cancer killers, which shows that these drugs, supposed to be potent inhibitors of protein A, actually aren't, and they likely have a much more complex polypharmacological mode of action.
If you stick to the classical way of doing drug R&D, it's complex enough. And there is the scientific complexity, and there is also the regulatory complexity. So do you think also one of the reluctance, beside the scientific complexity, one of the reluctance for one disease to go after multiple targets from the get-go might be also that we don't necessarily have even like a regulatory framework to handle this approach? And here again, maybe it's a very naive question because I'm by no means an expert on the regulatory approval of drugs, and especially of drugs targeting multiple targets. But when you were saying that's a question that immediately popped up in my head, that how will people even go about submitting all the information with different drugs targeting multiple targets at the same time for one indication?
Yeah. Yeah, I completely agree. I think that the regulatory context is one of the main reasons why the one drug, one target paradigm is still so well established. It's because simply the story, like we found one target, we designed one drug, and it solves the problem, is much easier to explain to regulatory agencies like the FDA. Then, okay, we need to modulate actually 12 different targets so that we have a strong effect on 4 different pathways. Also because the design of the subsequent clinical trials and all of the even before that, all of the preclinical evidence you have to produce is much smaller if your story is much easier. So I believe that we are voluntarily making the story simpler to avoid the complexities in the regulatory pipeline and in the amount of evidence that needs to be produced to put a drug on the market, which is a sad reality.
Yeah, completely agree. But now, so if we shift a little bit to nowadays, and I'm not going to talk about AI now, but let's say technology in general. One of the things that I think is remarkable with tech is there are, like, the traditional approach in science is to reduce, like the reductionist approach, to take a very complex system, to make it simpler so that you can study it. Like, if you go back many decades before, so you make the system simple enough so that you can make your in vitro experiment, then test it in animals, and then control all the parameters. And by definition, as you said, nature is complex, and the reality is that there are so many parameters, and when you reduce them, then you decide to omit a lot of those parameters. Keeping that in mind, so what do you think are the biggest challenge and opportunity now shifting and embracing polypharmacology and embracing the fact that biology is more complex than what we wanted to admit, and that there might be a better way of addressing diseases if you embrace this complexity and you say, look, I will not try to target just one target, but really look at the whole pathway and what is the minimum set essentially of points on this pathway that I need to inhibit or to stimulate or whatever, to modulate, to have an effect on the disease.
So first, you're I think completely right about reductionist thinking, natural way scientists work to also to create their publications. I think makes up for easier stories. Biological systems, if you pull one string, you're affecting the entire web. The problem is that previously we had no way of understanding the consequences of even inhibiting a single protein. Sometimes, like, we were, like, if you look at paracetamol is still one of the most widely taken drugs. It's still completely unknown mode of action, which is quite crazy when you think about this. But now there's the AI revolution. AI has the capacity to leverage tremendous amounts of data, which we have gigantic pathway databases that really record, like, I think, millions of reported single protein interactions or co-expressions or various co-occurrence of proteins. It's so much that the human brain cannot make sense of it. You cannot go through that and try to imagine all the potential effects that your drug will have by touching a single protein, by exploring all the links. Humanly, it's starting to be impossible. There's too much. But AI is perfect for that kind of work. And even when it comes to small molecule protein interactions, try to figure out exactly what is the potential of interaction between your molecule, and the whole proteome. Is something that AI can now solve. We have a lot of assay data, functional bioassay data, available to train AIs that can then have the power to predict this, meaning that we don't have to do in vitro large-scale assays anymore. We can just do the predictions and then do the minimal in vitro validations to double check that those predictions we've done are true. So if you have AI to explore drug-protein interactions and AI to explore all the pathways that we have already identified, it's much easier nowadays to say, okay, in the context of this cancer, modulating those three pathways at the same time has more potential than touching a single target somewhere.
So you mentioned all the data and all the data available, and I worked within pharma and with a lot of pharmaceutical companies, and I think there are a couple of challenges regarding data. Yes, there is a lot of data, but very often this data was produced before AI even was a concept. So the data is dirty, it's difficult to use. I worked on a number of projects where before being able to do anything clever with the data, people had to spend months locating the data, getting the legal right to use the data, and then to clean the data. So as a smaller organization, how do you do about acquiring the data or even generating the data? What type of data are you working with, and how do you get your hands on it?
It's a very interesting question. I'll start the answer with a small story. I was at a Congress not that long ago, and one of the first presentation started with this phrase, the data to solve drug discovery does not exist yet, which made me really laugh. Well, I agree that we need more data. We always need more data. More data is useful to train better and better models. If you look at what exists, as I said before, there are millions of bioassays reporting activities of tens of millions of proteins, of small molecules, against thousands of proteins. Yes, the data is highly heterogeneous, as you said, because the assays are different, the concentrations are different, the setups are completely different, it's in different labs, data is reported in different formats, but this data exists. The information, the signal is present, and it can be exploited. This is what we have been doing. And yes, our projects on that started with months of data curation, gathering, and some partnerships too, because there's a lot of publicly available data, but there are also companies that have a lot of privately owned data sets that can be leveraged. Regarding heterogeneity of the data, I will take the counterfoot to what people usually say. I believe that it's highly needed. If you want a model that has the capacity to generalize on a problem, you need highly heterogeneous training data. If you make the parallel with things like large language models, like ChatGPT, if you were to train it only on Wikipedia data, it wouldn't be capable of holding a day-to-day conversation with someone about the news or whatever. It would be like focused on a single type of outputs. To make it like resemble a human being when it talks, they have to train it on literally the entire internet. All the different types of data, discussion, text, books, and anything that's been written by humanity so far, which is highly heterogeneous when you think about it. We are doing the same thing, but with bioassay data. We gathered millions of very heterogeneous bioassay data that tell us about interactions between small molecules and larger objects that are proteins. We've put all that together, cleaned it up, and we now have a model that is capable of predicting any interactions between small molecules and proteins at scale. So if we want, we can screen thousands of small molecules against the entire human proteome, and we'll have a profile of interaction of these molecules against our biological systems. And then we can search through that for interesting things in the context of diseases.
Okay, so it's a good segue to actually, we've been talking about the challenges in the field and now data. So let's talk about your company more specifically and what you're doing. I was trying to find a list of all those tech bios, like companies that are using tech to address life science or biology problems. And every day you have 10 more. So it seems that it's one of these domains where all the tech companies are investing. And as you said, if we go back to the beginning of the discussion, it's full of good problem and ethical, quote unquote, ethical problem. If you solve this, you're doing good for humanity without trying to be too pedantic. This is an interesting mission, something important to solve. Back to my original question. So what is it that makes you different? So what is the thing that will differentiate Kantify from all those companies that are using tech to address life sciences challenges?
Interesting. So Kantify it's a tech bio with a mission to use AI to improve human health. This is something we decided 5 years ago, that from this moment onwards, we would only apply AI in the context of human health. We very quickly converged towards drug discovery. And what I believe differentiates us from the rest is the fact that we have created the so-called foundation model for small molecule protein interaction. And we use it to do target deconvolution, so to explain which therapeutic targets can be modulated for various disease types. We also have a very large focus on rare diseases. So we go where traditional pharma companies don't really go. We have dozens of partnerships with mostly academic, but also a couple of pharma and CRO companies in the context of rare diseases, rare cancer, and rare neuromuscular disorder diseases. We want to have an impact there. We want to go quickly towards those indications to have a maximal impact. We also have a couple of side pipelines on more classical indications like pain and non-rare cancers, obviously, but we have a special care for those rare indications. Our capacity to identify targets is, I think, what differentiates us the most from maybe other companies, because we have this Rosetta Stone that is Sapien Hit. Sapien Hit tells us about any possible interaction between molecules and proteins. If you think about it, it means that you can interpret any screening data results. So we work with partners who have phenotypic screenings. For example, they tested 1,000 molecules on a rare neuromuscular disorder. They saw that 10 had an interesting positive effect against the sick cells, but they have no idea how or why or what's happening inside those. So what we do, we take those 1,000 small molecules, make predictions of interactions with the entire proteome in presence, so all the proteome of the cell, and we would then deconvolute that to say, okay, the reason why those 10 molecules work is because they act upon a set of proteins that the others don't. And then we have the partner validate the targets, and once the targets are validated, we launch the drug discovery process, polypharmacological, searching for small molecules that will modulate at the same time several of these interesting targets validated. There are two successful cases I can talk about. Sarcoma and LGMD2, so limb-girdle muscular dystrophy of type 2, where it was exactly that. We had some source phenotypic inputs, which were screenings of molecules with the notion of readout. These have some effect that is positive and all the others don't. We made our predictions with Sapien hits over the entire proteome. We deconvoluted a number of targets we believed were playing a role in the response. The partners validated those on the in vitro model through some silencings first, and then silencing plus retesting the drugs that worked. And suddenly the drugs didn't work, which validates the role of the targets. And then the next natural step is that we took those targets and we designed drugs specifically to inhibit or activate all these targets at once. And the response in vivo is always positive so far when we do polypharmacology. In comparison to our single target projects, because we have a lot of collaborations on single targets, obviously, where customer partner comes in with a specific target of interest they would like to find hits for. We always manage to find hits, but the validation in more complex models like in vivo is always more problematic. As every time we focus on a single target, it's harder to get an in vivo response. That's why we are starting to believe in polypharmacology more and more, because we see way more positive results in the polypharmacological approaches than the classical single target projects.
But it's interesting because essentially you offer both. So you have the thing that you really believe in, the polypharmacology approach, but if someone comes to you and say, we have a target and we want to find hits, you'll still be able to help them.
Yes, exactly.
Yeah.
Exactly. We don't want to reject the whole structure of the industry. There has been some success cases of very selective single target drugs. If you think of maybe the statins, for example, and cholesterol or the proton pump inhibitors. These were cases of, okay, there was a clear target, clearly associated to a phenotype. If you find a modulator, you have the effect that you want. So,
and here, is there anything specific? Because I worked in the past on some program also where we had AI platforms where client will come to us with one target, and then we will have like a toolbox of approaches to find hits, whether we do a virtual screen or high throughput screening or DNA encoded library. And in some cases, like despite having relatively large library, sometimes we will not find anything. And the other thing also is that when people come to you with a relatively novel target, you can't really do virtual screening because to do virtual screening, you need to have some information that comes from the literature to be able to design your screen. Can you say a few words about your approach when someone comes with a target and what makes it so successful at finding hits and maybe different from some more conventional approach?
The point where I beg to differ is on the impossibility to screen virtually for novel targets. That is with classical approaches where models do not generalize. Sapien hits, we know, we have proof in silico, but also now in vitro, that it does generalize, meaning that even on a novel protein, it has the capacity to find modulators. We have several projects where we are first in class. There's like an ion channel that I've never seen any activator, despite large-scale screenings. In a single go, we screened 100-something compounds, and we found 11 activators. That's a hit rate. And the hit rate is always pretty much in this order of magnitude, if not higher, because activation is like a harder phenotype to achieve usually. Sapien generalizes because it has been trained on highly heterogeneous, but also extremely large data sets. It has seen thousands of proteins and millions of small molecules for training. And the coverage in terms of protein and molecule diversity is enough so that now when we present like a new protein for inference, the model still manages to recognize some features in it and to propose small molecules that are going to bind to it, modulate it, whether activation or inhibition. We have cases where the goal was to find, for example, silent binders of a protein that had nothing known for the design of PROTACs. It's sometimes more interesting to have silent binders that do not modulate any of the functions of the protein. And in those cases, like we had similar hit rates. We found 60 silent binders on 100 compounds screened, but we also accidentally found, I think, 20 inhibitors and a couple of activators. The reason why Sapien hit works this well is, and it's so interesting for novel proteins, is that it does not rely, for example, on known 3D structures. The way we featurize proteins is not dependent on the existence, the knowledge of the exact 3D structure of a protein. AlphaFold is extremely interesting, but we don't really rely on that kind of information. There are smart ways to featurize proteins nowadays using models like AlphaFold that embed all of the information a hit-finding model needs to know.
I'm a structural biologist by training, so I understand structure. And I like when I can see something and I see a pocket and I can put something in it. That I understand. What's the other approach then to, how did you say, to featurize a protein or a target to be able to find potential hits?
So here, I don't want to reveal too much, to be honest, but what I can say is that the biggest demonstration of AlphaFold is that the whole structure, the folding of the protein, is actually encoded in its sequence.
Yeah.
That's the conclusion of AlphaFold. If from the sequence alone you can predict the structure, it means that just in the sequence, the structure information is present. Obviously, it does so based on the learning on thousands of known structures attached to protein sequences, but the underlying conclusion is that if you use, for example, the embeddings of a model like AlphaFold, you have extremely representative vectors that you can use for various applications. And in this context, hit discovery.
So I'm sure it took some time to figure out all the things that you've told about. And often we focus on what works, but can you tell us maybe a bit more about the lesson that you learned along to come to where you are, the things that didn't necessarily work, and the thing that might be also interesting for people who are embarking into a similar journey using tech, AI to work on this type of very complex problem that is drug discovery and development?
I mainly have one idea for an answer there. It's to take orthogonal approaches. It's when you work in the field, usually you end up having a tunnel vision without really realizing, because you're just in. And when we got into drug discovery, we realized that everybody was talking about the same things. They were talking about docking, advanced docking techniques, AI-based docking techniques. And for that, you needed to have the 3D structure of the proteins known to just unlock the capacity for docking. So we looked at that and like, okay, based on the data we see existing out there, we just don't believe that it's a necessary step. And obviously, if you dig a little bit into docking, you see that more and more people say docking basically doesn't work amazingly well, rather. There are cases, very specific ones where it does, but Generally, docking hasn't had a tremendous impact on the creation of novel small molecules. So when we got into drug discovery, we didn't go towards docking or prediction of 3D structures of proteins or small molecules, whatever. We did our own little things. And now we have a model that is completely orthogonal to what everybody else seems to be doing. That doesn't rely on 3D structures, but also that leverages tremendous amounts of heterogeneous data to create a new foundation model that solves a very simple problem that is at the base of everything. Like, does a small molecule hit on a protein? It's quite simple formulated this way. So yeah, taking a novel orthogonal approach is, I believe, what's made us successful in drug discovery. And what's funny is that we did it completely by accident. It's just because we were experts, we were AI experts first. Coming from completely different fields, we used to tackle any kind of new data that we were given for different contexts. So we arrived, we looked at what data was available, we found those amazing data sets online. Like, it's crazy. I think health is really the sector that has the most data online. If you want to solve problems, go in healthcare. There's like entire databases of millions of data points reported by tens of thousands of researchers that you can leverage. And we saw that some of it seemed to be underused or underperceived, and we just made use of it.
In the past couple of years, AI has gotten more attention, more investment. When you think that you're at the peak of the hype, there is something new. There was the latest presentation from NVIDIA at CES where they were talking about physical AI. So I thought that this year would actually be the year of agentic AI, but now it seems that it's the two buzzwords that I'm hearing a lot is agent and physical AI / robots. So yeah, looking into the future, into your crystal ball. So what is it that you see that will be interesting, exciting, and have an impact on our field? And then we'll talk again in 5 years to see if you were right.
Let's see what kind of predictions I could make. For the pharma industry specifically, I believe that a couple of small players, AI first companies, are going to take over in the coming 5 to 10 years, because they'll have the capacity to multiply parallel pipelines. They'll have dozens of drugs in development very quickly for a very small cost compared to traditional preclinical pipelines. So I do think that in 10 years from now, we should be seeing the new drugs getting on the market from partial or full AI-based development pipelines, I would say. Right now, there's evidence that AI has accelerated preclinical development quite a lot, but I've read that the improvements are visible in phase 1, meaning we managed to detoxify the drugs that make it there, but the failure rate in phase 2, so for efficiency, is still the same. So admetox models predicting toxicity seems to work quite well. We can pass phase 1. Now the challenge is to make drugs that are actually efficient for the indications. With novel target discovery techniques that will identify the right pathways to modulate, this is going to change as well. So I think the success rates of phase 2 and 3 are going to climb in the coming 5 to 10 years, and that's for the good of everybody. And the other prediction I want to make, but it's mostly something I hope for, is that we'll start seeing drugs come out for rare disorders. With the prices of drugs, drug development going down, it's going to open the door for new markets. And so smaller markets, meaning rare diseases, should have a chance now to have their own drug development pipelines. So we'll see in 10 years if this happens.
These are great prediction, and yeah, I hope you're right, because I think it will be good for everyone if we can develop more drugs for cheaper, for more indications. So Nicolas, thanks a lot for this conversation. A very interesting take on some of the typical drug R&D problems. And as you say, I think the thing that will really stick with me is this, what you said about having an orthogonal approach. So essentially not trying to do the same as everyone, which maybe is one of the root cause of our problem in the industry. If people have been doing the same thing for the past 50, 60 years and it doesn't work, why continuing to do the same?
Yeah, Sounds about right.